The Planarian locomotor velocity test is a means for quickly assessing stimulant and possible withdrawal effects for a variety of biological modifying agents that might affect motility.
This assay is easily controlled and provides a straightforward and accessible method for generating reliable behavioral data in even the most basic research settings.
Since the Planarian nervous system has a centralized rudimentary brain, a bilateral symmetry, and analogous neurotransmitter systems, behavioral studies using these organisms offer preclinical insights into possible vertebrate responses.
Experiments can be further affected by physiological pathway modifiers, drugs, or even genetically modified planarians to provide insight into the associated biological mechanisms.
It is important to handle the planarians so that the transfers between containers do not stress them, and to standardize the grid line counts between lab members.
The best way to obtain consistent results from these behavior assay experiments is by seeing how experienced users perform them.
After setting up a planarian culture, feed the worms ad libitum on a regular twice-a-week schedule of chopped organic boiled eggs or blended organic beef liver for 1 to 2 hours. After feeding, remove any soiled water from the container and use a flat watercolor paintbrush to transfer any food debris and slime adhering to the container to a paper towel. Use fresh spring water and gentle swirling or agitation to dislodge the planarians and decant the worms into a clean container. Slide a wide-bore pipette tip under any planarians remaining adhered to the old container to facilitate their transfer to the new.